we learned about identity and similarity. So we'll learn about there is a qualitative statements called as homologs. So homologs, orthologs, paralogs and genealogs, four terminologies we use while comparing the sequences in qualitative approach. The definition of homologs is something like this: similar sequences in two different organisms derived from a common ancestor sequence. So in two different organisms, the sequences has sourced from two different organisms and they must be similar, and in evolutionary context they have a common ancestral sequence here, are called as homologs. And homologs can have subparts also, subclassifications, orthologs and paralogs here. Orthologs are two similar sequences in two different organisms that have arisen due to speciation event, and the function of the protein or the function of the sequences must be retained, functionality is retained here. And paralogs, similar sequences within an organism that have arisen due to gene duplication event here. So within an organism that have arisen due to gene duplication event and their function may be different are called as paralogs here. And genealogs, the genealogs terminology we don't use often here, very often. Two similar sequences that have arisen out of horizontal transfer events. That means they may not have direct evolutionary relation here. So maybe because of viruses or symbiotic organisms transferring one DNA segment from one organism to another organism here. So that is because of horizontal gene transfer events. So we'll see an illustration to understand this concept clearly. So let us assume that we have a early globin gene. So globin gene is a DNA segment which encodes for a globin protein. Examples for globin proteins are myoglobin, hemoglobin, ovalbumin. Like many proteins, majority of the proteins which are in cytosol or which are in our blood or plasma or serum, these are globular in nature and all are called globin proteins. So the gene which is corresponding to one of the globular protein, we call it as a early globin gene. So this early globin gene, because of some chromosomal aberrations or some kind of events at the level of DNA sequence, the gene has been duplicated, and the early globin gene now it formed into alpha chain gene and beta chain gene. So it has become duplicated and it might have undergone some kind of changes so that they have separate identities, alpha chain gene and beta chain gene here. Okay, so this one, so from this organism, from this species where this preliminary gene sequence has existed, so three different species has been arisen here. So frog, chicken, mouse. So three different organisms from a common ancestor here. So this alpha gene can be present in frog, beta gene also can be present in frog, and all of these two genes can be present in frog, chicken, mouse. So different organisms. Now you see here gene duplication happened here, and because of gene duplication, so you have two different kinds of genes are there, and then three different organisms. So now the alpha gene which is present in frog, chicken and mouse, three different organisms, and since it is alpha, same function, so you can call them as a ortholog. And coming to the alpha and beta genes in mouse, so both are in same organism, their function is different but they have common ancestor, they have some sequence similarity, so those are called as paralogs here. So orthologs and paralogs together defined as homologs here. So like this we can relate the sequences as orthologs, paralogs, and or broader sense homologs based upon the gene duplication and speciation event here. So when something we say two sequences are homologs, that means they are having same function and similarity more than 25% or 70% for DNA or proteins, vice versa here. So these are the definitions here. And coming to the another definition. So while doing the sequence alignment, we can do two kinds of approaches or two types of approaches. The approaches are called as global alignment and local alignment. Two different approaches are there. In global alignment, what we do is we take first sequences start and second sequences start, and also you take first sequences end and second sequence end, and you try to compare as much as possible to the total length of two sequences here. So during the process you can insert some kind of gaps in the two sequences and try to adjust the lengths so that they have similar length and they are properly aligned from beginning to the end. That is called as global sequence alignment here. And while local alignment, it doesn't care about lengths of two sequences, it go for exact matches of two sequences. One can be at the beginning of first sequence and second one can be at the end of the sequence here. So here somewhere the match can be here or within the middle or also in the cross orientation. So local means you don't care about the lengths here, only you take the exact matches irrespective of the positions in two sequences. So like this, two kinds of alignments can be done, global and local alignments here. Okay, so thank you for listening to this session here. So in this session we learned about the definition of sequence alignment and why sequence alignment is done, and what are qualitative and quantitative approaches for sequence alignment. So we learned about similarity, identity, homologs, orthologs, and what are different types of alignments here. So we'll discuss about these things in lab sessions while performing the experiments also.